satellites from space. There was a recent algal bloom in the North Atlantic that was actually the size of the state of Alaska. And it was just made up of many, many trillions or quadrillions of copies of this little individual cell. So those are all eukaryotic cells. This up here is Escherichia coli, who is a bacterium that is much beloved of laboratory scientists. And we'll talk about that actually quite a bit more. But this is a bacterial cell on the same footing as all of the other cells. So I'd like to just briefly mention the major distinction in cell biology based on cellular morphology between prokaryotic cells and eukaryotic cells. So we, other animals like whales, including invertebrates like the fruit flies, and then also plants like this redwood tree, we are eukaryotic organisms, which means we have all of our DNA enclosed within a membrane-bound compartment, or the nucleus. Now that is a choice that our ancestors made on the order of between 2 and 2.5 billion years ago, but it's not a particularly important choice, and it's certainly not the only way to be. All of the rest of the cells on the planet are prokaryotic, which means before the nucleus. And these guys have their DNA just in the cytoplasm, along with the ribosomes and all of the other metabolic enzymes and all the functioning parts of the cell. Prokaryotic is a commonly used term, but it's actually kind of an old term. And we appreciate now that the wide variety of prokaryotic cells can really be better described as being divided into two large kingdoms. And those are the bacteria and the archaea. And I'm going to focus today mostly on the bacteria because those are the ones we understand best, but I will say a few words about the archaea here and there. Now, the easiest way to tell the difference between a prokaryotic cell and a eukaryotic cell, if you're looking down the microscope, is simply size. Prokaryotic cells, like bacteria, tend to be fairly small, and they tend to be fairly simple. They'll have shapes like rod shapes or spherical shapes or sometimes little spirals. But if you look down the microscope like at pond scum and you see something that's got, big long whips or is swimming around or is eating something else or is sort of changing shape and oozing, that large thing that's morphologically complex is going to be a eukaryote. So although they're small and they're morphologically simple, they greatly outnumber us. In our human body, it's probably about 10 to 1 prokaryotic cells to eukaryotic cells. On the planet, it's very difficult to actually take a good census of the total number of cells, but it's estimated to be between 1,000 to 1 and 1,000,000 to 1 that prokaryotes outnumber eukaryotic cells on the planet. And as I mentioned, even among the eukaryotes, most organisms are microscopic. Now, although these cells are very different in terms of their appearance, there are a lot of things that they have in common. And in particular, I know Gil Chu introduced you to the central dogma of molecular biology, that is the concept that DNA is replicated, DNA is transcribed into RNA, RNA is then translated into protein. That fundamental three-step mechanism for the expression of genetic material is shared by all cells on this planet, all the bacteria, all the archaea, all the eukaryotes as well. And one thing that's actually very convenient about that, if you're interested in surveying the identity and the location of cells, for example, of bacterial cells, is the fact that because all cells on the planet work fundamentally the same way as far as the central dogma goes, that means you can count on every cell having certain components in common. And the one that's actually turned out to be the most useful for the kinds of studies that I'm going to be describing is the fact that all cells have ribosomes. So ribosomes, remember, are the big machines that take the messenger RNA and then translate the triplet code in the messenger RNA into a protein, and that protein can then go on to have biological function. And the ribosome itself is an incredibly elegant, complex, beautiful piece of biological machinery. It's extremely large. It's 25 nanometers across, which makes it about 50 times bigger than an average-sized protein. And it's not a single protein. Instead, it's a complex that's made up of many, many components or substituents. And of those components, a lot of them are in fact proteins. So for example, the bacterial ribosome that does the work of translation in a bacterial cell contains 55 proteins, but it also contains 3 large RNA molecules. Now these are specialized RNA molecules that never get translated into a protein. Instead, their sole function is to make the structure of the ribosome that does the translating of other RNAs. If we look in eukaryotic cells, they also have ribosomes, but their ribosomes are a little bit different. They tend to have more proteins, they're a little bit fancier, and they also have 4 RNAs instead of 3. But you can pretty much line up the RNAs from all the eukaryotic cells with the RNAs from bacterial cells and see how they're related to one another. Because of this, because of the fact that every single cell on the planet of every possible different kind has ribosomes in it, and specifically has ribosomal RNA in it, if you want to make some kind of measurement or some kind of calculation about how closely related any two cells are, one very convenient way of doing this is just taking the sequence of the ribosomal RNA and comparing that one to another. So for example, if you were to take human ribosomal RNA and compare it to chimpanzee ribosomal RNA, it would probably be almost identical. If you were to compare either of those to the ribosomal RNA from a dog, you might see a few changes. If you were to compare either of those to the ribosomal RNA of a fruit fly, now you begin to see some more changes. To a redwood tree, yet more. To a single-celled organism like Tetrahymena, the pond scum, you'd see more. And if you compared it all the way to a bacterial cell, you'd see even more changes. But the point is, they're still similar enough. You can still line them up, and you can count how many changes have taken place among all the different cells on the planet. And this is absolutely astonishing. And this has been used in a lot of very creative ways by a lot of scientists to try to understand how we can look back in time and see how evolution has occurred and see how everybody is related to everybody else. And one of the early examples of this was a really spectacular piece of work that was done by Norm Pace and his colleagues at Indiana University in the early 1990s, where they just went around taking various samples of different things, from things like hot springs in Yellowstone National Park, water out of the ocean, soil, deep ice core samples from Antarctica, and then just measuring the number of changes that had happened in ribosomal RNA from all the ribosomal RNA they found in all the cells in those samples and comparing them to everybody else. And the really profound thing that was proved by that series of experiments is that every single cell on the planet, including all of your cells and all of the bacteria and everything in between, they all are descended from a single common ancestor. That is, we can line up all of these ribosomal RNAs and we can see how they're all related to each other, and by counting the changes, we can see how they diverged. So when we're looking at this image, and this is a kind of tree that I'm going to be showing you a lot of, the length of the branches in the tree shows you how similar or how different two different sequences are. So for example, if you see something like this little branch up here, where you see these three branches that are connected with short lines, that means those three sequences are all fairly similar to one another, and therefore perhaps diverged from one another relatively recently in evolutionary time. If you look at things that are related only by very long branches, so this here, compared to this here, the longer the distance that you have to travel on the tree before you get to the other thing, the more different the sequences are from one another. So this is sort of a convenient way of classifying very large numbers of sequences to say sort of what's the pattern of diversification. Now just to orient you here, this tree from a publication from Norm Pace's lab from 1996 shows all of the three major domains of life. Where down here are the eukaryotes in this branch, here are the bacteria up in this branch, and here are the archaea over in this branch. And this kind of work was one of the things that established the fact that archaea and bacteria, although they look alike to us, are really very different from each other, and in fact have been diverging for just as long in evolutionary time as the eukaryotes have been diverging from the bacteria. So if you compare all of the cells up here, the archaea and the bacteria, again, they're wildly divergent. They've been evolving away from one another for between 3 and 4 billion years. But they look very similar, and in fact what they've concentrated on is diversifying their metabolism. So we have extremely exotic metabolism going on in these cells. These were the cells that invented photosynthesis. In fact, there's never been a eukaryote on this planet that can do photosynthesis. And the only way that things like trees and plants are able to do photosynthesis is because a long time ago they captured a bacterium, they captured a cyanobacterium and domesticated it and had it turned into a chloroplast. But the exciting metabolism that goes on there that changes light into sugar, that was invented by a bacterium. We have bacteria that are able to eat rocks. We have bacteria that are able to generate methane. All of that complicated metabolism that's only done by prokaryotes. Eukaryotes are extremely boring as far as their metabolism goes. So what have we got going for us? Arguably we rule the planet. I mean, here we are. We have civilization and all those kinds of things. The way that we're able to have civilization, even though we have extremely boring metabolism, is that we're very good at doing morphology. We're very good at making large cells, cells that do fancy things, cells that swim around, and particular cells that like to pile up to make big multicellular organisms. But if you look at the multicellular organisms, everything you can see with the naked eye, that basically comes down to animals, plants, and fungi. And here on this tree of all the cells on the planet, those are right over here. So this branch here, Homo, that's us, Homo sapiens, and this branch actually represents all of the animals. And I don't just mean all of the mammals, and I don't just mean all of the vertebrates. I mean all of the animals. So fruit flies are indistinguishable from humans on this image. This here, Coprinus, is a mushroom. That represents all of the fungi. And this here, Zea, is corn, and that represents all of the plants. So at least as far as our ribosomal RNA is concerned, everything you can see with the naked eye is just a tiny, tiny twig off of one side of the giant tree of life. And so what I'm going to focus on here now is the bacteria. And I hope at this point I've convinced you that bacteria are interesting and worthwhile and at least should be considered as co-inhabitants of the planet along with us and co-inhabitants of our bodies. Yes?
Clarify once again, back to the Luca, when everything was that, how did it evolve up here, up there? So you captured bacteria, how did it capture it? That's all it was.
Yeah. So the question was, how did the Luca, the last universal common ancestor, how did that evolve into all of these different branches? So this is extremely controversial. And I don't want to give you the impression that Luca was a single cell. It's not like there was one cell that was located at some location that then gave rise to everything that lives. Luca was a population of cells. And I'm going to tell you now what I believe is the most likely explanation, but as I said, this is very controversial. But I believe what probably happened is that there was a period of evolutionary history where there were a lot of cells on the planet that were very enthusiastic about swapping their genetic material back and forth. They shared their genetic material among each other. So it was a population where everything was held in common, sort of communist cells, if you will. And then there was some point in evolutionary history, and again, the numbers are not clear, but it was on the order of about 3 billion years ago, maybe 3.5 billion years ago, where some cell got the really clever idea that it was just going to keep its DNA for itself, and it wasn't going to share it with anybody else. And that has been referred to by Carl Woese at University of Illinois as being the Darwinian threshold, which means that once you get to be selfish about your genetic material, then if you make a mutation that's good, then instead of giving it to everybody else so everybody benefits from it, you keep it from yourselves and then only your offspring will get it. Now, once that happens, then you can begin to diversify. Because if the DNA that you make, you mutate, you adapt only goes to your offspring, then that gives you individuality and it gives your lineage some sort of separate identity from everybody else on the planet. So the sort of general consensus is that before this Darwinian threshold was passed, there was a huge amount of evolution that actually gave us DNA as the genetic material, that invented RNA polymerase, that invented protein translation and invented ribosomes. But all of this really complex evolution was all taking place under a context where you couldn't have separation of lineages, you couldn't have natural selection. And then after that happened, then you were able to diversify. Question?
How did that practically happen? Was everything having sex with everything else?
Well, so sex is a very late invention. And it's not actually the best way to swap genetic material around. In fact, when we talk a little bit later about what the bacteria do instead of sex, you're going to be jealous because it's much, much more efficient. So sex is only, that's only if you're diploid, it's only if you're a eukaryote, and really you shouldn't be that proud of it. So it was much more just that genetic encoding of information in the form probably of DNA and RNA was able to be freely shared. Now, there's a lot of controversy as to what that really means. So, for example, right now, every cell on Earth is surrounded by a membrane, and that membrane keeps everything on the inside. And arguably, that is how you distinguish inside versus outside and self from non-self. Now, there's very good arguments for why, even before the Darwinian threshold, there still must have been membranes around those cells. There's metabolic arguments that I can go into later, but I think it's pretty clear there were membranes. So if there were membranes and things were physically separate, why were they still able to swap things around? I don't think anybody has a good answer to that. I can't tell you how much money I would give to be able to go back 3 billion years and actually look at what was floating around on the planet, at that time. I think any scientist would do that, but we just don't know. And, the problem really is there could have been a lot of burgeoning life. I mean, there were a lot of cells that any modern biologist would recognize as being cells, but they're all gone now because once somebody made the decision to be selfish and keep the DNA, it could figure out how to do things like eat the other cells. And so all those other guys, they're gone. All we have left is the descendants of these lineages that decided to be selfish. And so at some point around then, it broke out into these 3 major branches, and then they diversified from each other. But exactly what happened at that transition, that's a very hard question.
So were these cells promiscuous or incestuous?
Oh, well, okay, so the question was, were the cells promiscuous or incestuous? I would argue that if you're a part of a collective, that there is no distinction between promiscuity and incest, so both.
Viruses, what roles do viruses